Hello everyone. Namaste to all my dear aspirants, my dear doctors, surgeons, gadgeted officers. In this video, I will explain each sub-stage of Prophase 1, Leptotene, Zygotene, Pachytene, Diplotene, Diakinesis. As if we are going to learn watching this love story, the love story of two homologous chromosomes. So imagine two long lost lovers finally meeting after a long time. Our story today is about two homologous chromosomes who come together, pair up, share secrets, and sometimes even exchange gifts. So, we will learn stages of Prophase 1 in a fun way. Yes, you heard it right. This is not just a love story, but Prophase 1 of Meiosis 1, one of the most important stages in cell division. Many direct questions in NEET and AIIMS have been asked from this single stage. So, stay tuned till the end, where we will solve PYQs together. So mnemonics for order of stages. Little zebra plays drum delightfully. Little zebra plays drum delightfully. L for leptotene, Z for zygotene, P for pachytene, D for diplotene, and then D for diakinesis. Mnemonics for order of stages are little zebra play drum delightfully. So key events of Prophase 1, Meiosis 1, which we are going with the help of homologous chromosomes. Love story. First to leptotene. Leptotene chromosome begins to condense. It is a very important point. In leptotene stage, chromosome begin to. It is a just important points we have to memorize. Just we have learned the mnemonics. It is an important point of Prophase 1 of stages, 5 stages. So leptotene chromosome begin to condense and become visible as thin threads. Each chromosome consists of two sister chromatids. Chromosome start searching for their homologous partner. So the story of two homologous chromosome will start from leptotene stage. Zygotene homologous chromosome start pairing. They got the partner and synapses occur. So zygotene stage, homologous chromosome got partner and synapses occur. So synaptical complex starts forming between homologous chromosomes. Synaptical complex in zygotene stage means there is affection started between the homologous chromosome in zygotene stage because they got the partner. In Pachytene stage, they start the crossing over. Pachytene P4 and then crossing over. So homologous chromosome are fully paired, synapsis is completed and crossing over take place, genetic recombination occur where non-sister chromatid at chiasmata. Exchange of genetic material introduces genetic variation. In Diplotene stage, synaptical complex dissolved. So they are now two lovers are going to separate. So the synaptical complex, that complex of attraction, affection, love will disappear. So synaptical complex dissolved. Homologous chromosome begin to separate but remain connected at chiasmata, site of crossing over. Chromosome becomes thicker and shorter in diplotene stage. Then diakinesis, fully they are separated, chromosome are fully condensed. So chiasmata shift towards the end of chromosome. Process is known as a terminalization. Nuclear membrane nucleus disappear, spindle apparatus begin to form. So these are the main stages which we are going to learn with the help of love story of homologous chromosome. So early stage of condensation, chromosome condensation is leptotene and zygotene. Zygotene, it is a leptotene is first stage of Prophase 1 where chromatin fibers begin to condense, become visible as thin thread-like structures under a microscope. Zygotene is further condensation occurs as homologous chromosome begin to pair up and the process is known as a synapsis. Formation of visible chromosome is totally visible in Pachytene stage, after Pachytene stage and onwards. So Pachytene stage, the chromosome become thick and visible. They are composed of two sister chromatids. Two sister chromatids joined at the centromere, joined at centromeres, forming a tetrads. They are forming a tetrads because bivalence, and it's four chromatids, so it becomes tetrads. Diplotene and dikinesis states condensation continues, leading to the final condensation of chromatin fibers during the last stage of prophase 1 dikinesis. So we are going to learn in what phase, the question will come, in what phase does chromatin condensation occur? So chromatin condensation occur in prophase phase of both mitosis and meiosis. Mitosis and meiosis, prophase phase condensation of chromosome chromatin occur. So it is a primary period when chromatin condensation occur, during which the DNA and protein histone that form chromatin are tightly packed. DNA and histone protein that form chromatin are, chromatin is made up of DNA and histone proteins. The chromatin are tightly packed and coiled to form visible compact chromosome that are ready for cell division. Now it becomes ready for cell division. It becomes compact chromosome. So what happens during chromatin condensation in prophase? So it uncoiling of DNA take place, chromatin fiber formation take place, compaction, it compact chromatin compact into chromosome, visibility under a microscope. So why it is important? So we are going to learn this one. So first point is what happens during chromatin condensation in prophase? Uncoiling DNA. The long strand of DNA are wrapped around histone proteins, which are called nucleosome. For this also I will make separate video. So the long strands of DNA are wrapped around histone protein and called nucleosome. Now next stage is chromatin fiber formation. Multiple nucleosome coiled together to form a thicker chromatin fibers. Next, what happens during chromatin condensation in prophase? It compact into chromosomes. The chromatin fibers further coil and condense into short, thick, rod-shaped structure known as a chromosome. It is visible under a microscope. The significant condensation, because it becomes thick, so it is visible under microscope, under a light microscope for the first time. Why it is important, this condensation, why it is required? Because it is important for proper segregation, for separation. The process of condensing chromatin into chromosome is important for accurate separation of genetic material into daughter cell during any phase, either mitosis or meiosis, so occur in prophase, in prophase stage. It help in protection also. The highly condensed form protects the DNA from being damaged during cell division. So it's compact. It is important for condensation. It protects the DNA. When there is a cell division take place, the DNA will not be harmed. So why it is important? After prophase, what happened? After prophase, telophase, when it recondense, a chromosome later decondense and uncoil back to the thread-like chromatin form as nuclear envelope reforms. It is a PYQ. When chromatin condensation in the prophase stage of mitosis, meiosis, and when it decondense, it decondenses telophase stage and uncoil back into thread-like structures, thread-like chromatin form as the nuclear envelope reforms. So these are about the interphase stage, which I will explain next video. Leptotene, Zygotene, which I'm going to explain you. Leptotene stage, Zygotene stage, Pachytene stage, crossing over in Pachytene, bivalence with four chromatids, nuclear membrane disappears in Diplotene stage, chiasmata, crossing over, asteroids formation in dikinesis. I'm going to explain now. So prophase 1 of meiosis 1 is more complicated and prolonged as compared to the similar stage of mitosis. So it consists of 5 stages: leptotene, zygotene, pachytene, diplotene, dikinesis. So homologous chromosome form bivalent tetrads and crossing over occur between non-sister chromatids. In this, we are going to learn. So prophase 1, leptotene condensation of chromosome attached to the nuclear membrane via telomeres end of the chromosome. Zygotene synapses begin with the help of synaptical complex forming between homologous chromosome. Pachytene crossing over of genetic material occurs between non-sister chromatids. Diplotene synapses end. Diplotene, the love that attraction will end. between the two homologous chromosomes. Diplotene means love and attraction between the two homologous chromosomes will reduce now. So synapses, and with the disappearance of that complex, synaptoidal complex, so the homologous chromosome remain, but remain attached at chiasmata. It remain attached at chiasmata. So diakinesis chromosome fully condense and nuclear membrane disintegrates prior to metaphase 1, broken down. So we are going to learn the story in the leptotene stage. Leptotene stage in the first, which is the first stage, of prophase 1. Leptotene is the first stage of prophase 1. Is like mitosis. It is like mitosis. Before prophase starts, genetic material, the DNA is in the form of thread-like structure. Before before the process of prophase starts, either in mitosis or meiosis. Now we are going to learn the meiosis. So before prophase starts, genetic material, the DNA is in the form of thread-like structure. DNA is in the form of thread-like structure, which is called chromatine. Chromatine. As the cell division start, it condenses to form chromosome. It condenses to form chromosome. So when a love story begin to form in couple, so when a child attain puberty, there is some complex will arise. So when a love story begin to form in couple, they love to put their heads into lap of each other. This is the first phase of love story. So this is the reason it is known as a leptotene. Leptotene. They are putting their lap of each other. So actually lap means thread. Lap means thread. Thin and thread. So initially the chromatin is thin and thread like, and then condensed to form chromosomes when child attain puberty. So in leptotene, condensation of in leptotene, thin thread like chromatin start condensing, compacting, occur to form, it form chromosome. Start forming chromosome. So mostly condensation occur in leptotene stage. So leptotene is also known as leptonema. Leptotene stage is also known as a leptonema. It is the first stage of meiotic division following the interphase. So chromatin fibers of interphase nucleus get shortened, elongated. Chromosome become clear, visible. So each chromosome appears to be longitudinal single. So look at an image, longitudinal single. The chromosome bear a series of dense granules which are called chromosomes. So in animal cell, one of the two centriole pairs begin to move to the opposite sides. One of the two centrioles begins to move to the opposite poles. So both the centriole pair and its astral rays together constitutes ester. In animal cell, points should be noted. One of the two centrioles pairs begin to move to the opposite poles. Look at an image. Begin to move to the opposite poles. But the centriole pair and its astral rays together constitutes aster. So these are two sets of chromosome in a cell undergoing meiosis. The chromosome number is diploid. So what is the chromosome number in leptotene stage? It is diploid. Condensation of chromatin into chromosome. So aster formation begin during early stage of prophase or early prophase of both mitosis and meiosis. Points should be noted. Aster formation. So what we have learned, the chromosome bear a series of dense granules which is called chromosome in animal cell. In animal cell, one of the two centriole pairs and begin to move to the opposite side, and both the centriole pair and its astral rays together constitutes centriole astral rays together constitute aster. So aster formation starts during the early stage of prophase or early prophase of both mitosis and meiosis. When centrosome begin to move to the opposite poles, it radiates microtubules. So look at an image, microtubules. It radiates when it moves to the opposite poles. It radiates microtubules forming aster rays that help organize the spindle apparatus. It helps to organize the spindle apparatus. Spindle apparatus. So what are the key characteristics? Aster are found in animal cell but are generally absent in plant cell. Very important point. These aster are absent in plant cell, which do not because centriole are absent in plant cell, but it still form a spindle apparatus. It forms spindle apparatus in plant cell, but aster are absent in plant cell. It's again PYQ. Okay, examination NEET PYQ, AIIMS PYQ. Now, microtubule based aster are composed of microtubules. Look at an image. Asters are found in animal cells. So, when it moves to the opposite poles, it radiates microtubules. These are called microtubules. Opposite poles, microtubules. So aster are composed of microtubules, which are dynamic protein structure, which are involved in the movement. What are the function of the aster? Aster are composed of microtubules and help in the movement and organization of chromosome. So early phase of cell division, aster formation initiate in prophase, and it is a preliminary step for overall formation of mitotic meiotic spindle. Very important point. Again, it becomes PYQ. It becomes PYQ. Aster formation initiate in prophase of mitosis meiosis and is a preliminary step, starting step for the overall formation of mitotic or meiotic step. So it helps in the formation of meiotic mitotic spindle. Clear? It is also one PYQ. Now aster formation located image, and now move to the zygotene step. Then as the love bond, because the love bond in between both homologous chromosome becomes stronger, they become close to each other, they pair up with each other, and as they pair up with each other, one of them lie on the top of the other. So when they do so, so a special bond is formed between them, and there is not just a bond of love, that is a bond of proteins. So when there is a love increase, so there is a bond of proteins initiated between both couple, between both homologous chromosome, and it is known as a synaptical complex. So complex of protein is associated between the homologous chromosome. So a protein complex, look at an image, it is a homologous chromosome, and protein complex is arise between the love bond between two homologous chromosomes. Synaptical complex is a protein complex. Again, it is a PYQ, and it is start in Zygotene stage. Start in Zygotene stage. So a protein complex is found between the maternal chromosome, and one chromosome is coming from father and one is from mother. So a protein complex, a complex of protein is formed between maternal and paternal chromosome of each homologous chromosome pair, and this complex is known as a synaptical complex. So it started in the, because the love between the both the homologous chromosome will going to be deepened. So complex arise, and that complex arise in Zygotene stage of prophase 1. Now it means that it is a synaptical complex between the two homologous chromosome, complex of protein. Nemal means thread. So this thread like synaptonemal complex. So nemal means thread. So this thread like proteinaceous complex is a synaptical complex, and this process of pairing is called synapsis. Pairing is called synapsis. So synapsis start in Zygotene stage. So this process of pairing of paternal chromosome, maternal chromosome, this is called synapsis, and it is demarcated by presence of synaptical complex, and because they are pairing and they are loving each other, we call it Zygotene phase. We call it Zygotene phase. Zygotene phase. Zygo means to pair up. Pair up with each other. So complex arise between the two homologous chromosomes, synaptical complex, and synapses occur. Love become more deeper. Zygotene stage, the homologous chromosome come to lie side by side and get attached laterally due to the development of nucleoprotein between them. So that nucleoprotein is synaptical complex, pairing of such that the gene of the same character present on the two chromosome come to lie exactly opposite. Pairing happen between the two homologous chromosome, maternal and paternal chromosome, such that the gene of same character present on two chromosome come to lie exactly opposite. It lie opposite. The pairing of homologous chromosome is called synapsis, and important point, synapsis is also known as syndesis. Syndesis. Again, it PYQ. Synthesis. So, account of synapses, chromosome form pairs or bivalent. The number of bivalent is half number of the total chromosome. Again, it's PYQ. The number of bivalent is half number of total chromosome, haploid number of chromosome from both parents. So homologous pair of chromosome nucleus centromere and its a formation of bivalent. Look at an image. Bivalent, one from maternal of chromosome and one from paternal chromosome. One from mother, one from father. So bivalent, formation of bivalent. So during synapses, what happened during synapses? We are learning zygotene. Zygotene means coming together, loving each other. So zygotenes during synapses, homologous chromosome pair up to form bivalents, which is called tetrads. And the total number of bivalents formed is half the total number of chromosome in the cell. PYQ again. Each bivalent contains two chromosomes. So the two chromosome means one from mother and one from father. So the number of bivalent is always the half the number of chromosome. So synapsis is the prophase, synapsis, key of synapsis, which is the process where homologous chromosome pair up during prophase 1 of meiosis. If it's a question is coming, which stage of prophase 1? In which stage, sub-stage of prophase 1, then it is zygote. Homologous chromosome, a pair of homologous chromosome that have the same gene, one inherited from. Homologous chromosome means it has same genes, one inherited from mother and one from father. Bivalent means the complex formed by two synapsed homologous chromosome. Tetrad means another name. It is also bivalent is known as a tetrad because it consists of two chromosome. Means it consists of two, two chromatids. So bivalent. Look at image, one from father, one from mother. So it is called bivalent. It is a pair of homologous chromosomes. So it is consists of two chromosomes. Therefore, it consists of one chromatids, one chromatids, four. So it becomes four chromatids. So this is the reason it is known as a tetrad. Tetrad. Otherwise, it is a bivalent, also called bivalent because one chromosome is coming from father, another is coming from mother. So homologous chromosome often form from each parent, one from mother, one from father. So find each other and align side by side through a process called synapsis. So love affair between both the couple, between the homologous chromosome will depend. The paired structure is known as the bivalent. So very important point now again PYQ. If the diploid cell has a total of n chromosome, it has n by 2 pairs of homologous chromosome, haploid. Since each bivalent represent one pair and there will be n by half bivalents, n by 2 bivalents, making the number of bivalent exactly the total, half number of chromosome, total number of chromosome. Clear? I explain it. So again, very important point. So chromatids, centromere, homologous chromosome. Sister chromatids, one from father, one from mothers, and then synapses occur between the both. So it becomes bivalent because of presence of two chromosomes, one from mother, one from father. It consists of four chromatids. This is the reason it is known as a tetrad. After synapses. Now we move to the third stage, Pachytene. Pachytene. Now what happens when a couple lie on top of one another? So, and while doing something, some material is exchanged in between them. So it's when they love each other. So there is a exchange of genetic material take place. So there is some exchange of genetic material. So in Pachytene stain, they are packed in love. They are packed in love. So they exchange of genetic material take place between the both the couple. Exchange of genetic material take place. So crossing over take place. CO take place. Crossing over take place. Cop. So Pachytene stage packed with love, affection. So what happened? There is a crossing over, exchange of genetic material take place. So in this phase, which crossing over take place, exchange of genetic material take place in packeting. So the couple are packed with each other to do something. Pachytene is also known as a pachynyma. So the paired chromosome or bivalent become short and thin. It becomes short and things. The chromatids of each paired chromosome slightly separate, and then there is a fight arises between both the couple, between both the homologous chromosome. So it's slightly separate and become visible. Thus, each bivalent or chromosome pair is made up of actually 4 chromatids, 2 of each chromosome. So 4 chromatids tetrads and 2 of each chromosome bivalent. So each group of 4 chromatids is called tetrads. During packeting, there occurs exchange of these corresponding segments and segment between non-sister chromatids of homologous chromosome. The process is called crossing over. Exchange of genetic material take place. Crossing over take place by breakage and reunion of chromatid segments. So non-sister chromatids of homologous chromosome in between the crossing over take place, exchange of genetic material take place. So one from mother and one from father. So this is the reason non-sister chromatids of homologous chromosome. Synapses of homologous chromosome crossing over. So after crossing over, the 2 chromatids of chromosome become dissimilar. Homologous chromosome, one from mother, one from father, crossing over between non-sister chromatids, crossing over between 2 non-sister chromatids, one from mother, one from father. So exchange of DNA between non-sister chromatid take place. So the love deepen between the 2 homologous chromosomes. So these are sister chromatids, and exchange of genetic information take place. Look at an image. So it's a called crossing over. Crossing overs, exchange of genetic information take place, and in this stage what is happening, chiasma formation take place. That's both are combining each other. Except structure is known as a chiasma. A newly appeared gene in is known as a recombinant. So homologous chromosome aligns. Look at an image. ABC, 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 small is this one, small ABC, small ABC, and there is a exchange of genetic material to take place. ABC, 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 these are the non-recombinant chromosome. And these are recombinant chromosome, and there is exchange of genetic material take place. The difference between the new genes and non-recombinants, nothing has changed. No gene has changed. No variation has occurred. So crossing over take place always between non-sister chromatids, one from mother and one from father of homologous chromosome. This result in exchange of genetic material between between two homologous chromosome. The crossover events are the first source of. This is the reason the genetic variation occur in the sexually reproducing organism. Genetic variation in the nuclei produced by meiosis. So diplotene stage, Pachytene synapses occurs, crossing over take place in Pachytene stage, synapses in Zygotene stage, and Diplotene stage. What happened after a couple has done something, they will have to start fighting each other. Some misunderstanding has arisen, so they decided to separate from each other. And Diplotene. So diplo means they are diplomatic. They are diplomatic to each other. So they have fighting with each other. Those start separated with each other. So they cannot all time remain attached with each other. So they must detach from each other, separated from each other. When they separated from each other, so what happened, because they are decided. So when they are a complex of synoptimal complex will vanish. There is no complex because they are started fighting in diplotene, diplomatic behaviors. So this is the reason they start fighting. They are not thinking of each others and they started fighting. So diplotene stage, synaptical complex, that complex protein is disappear. So diplotene stage, di means two. They now become two. Now they become two. So they are now separated. Di means a single entity. So they paired up in zygotene. They paired up in zygotene. Synapses happen and crossing of genetic information take place in pachytene crossing over in pachytene, and now they are different entities because they are fighting each other. Homologous chromosomes. So diplo means di. So it becomes single entity, one. So synaptical complex disappear. So, but remember, they are still attached, but they have not taken any legal things. So they are still attached at a point. At that point is known as a cross-like structure, that is a chiasma. They are still attached at diplotene because they have not gone for any legal process to be separated. So they are still attached at one place, chiasma. Chiasma. So chiasma are what? The child of these homologous chromosomes. So they are still attached at this point. Chiasma means cross. So chiasma formation occur in diplotene. Diplotene. So there are usually more than one chiasma, that is called chiasmata formation. The homologous chromosome are called bivalent and tetrad, we understood. So homologous chromosome are bivalent as well as tetrads. So bivalent means chromosome and tetrad means chromatid. So homologous chromosome, they are still attached at chiasma, bivalent tetrads, and then after it's they are separated into daughter chromosome after meiosis 1. So diplotene stage, diplonema, the nucleoproteins, this is the other stage when the 4 haploid is forms after meiosis 2. Meiosis 2. 4 haploid gamete formed after meiosis 2. Diplotene stage, the nucleoprotein fusion complex of the synapse chromosome dissolve because they start fighting, so complex dissolve. Diplomatic behavior, so they are separated, they are now in a single entity. So this is the reason the complex between the homologous chromosome will disintegrates. They are start detaching with each other. So complex will dissolve. So synaptical complex dissolve. Synapse chromosome dissolve. The homologous chromosome starts separating except in the one region, that is a cross region, that is a chiasma. The point of attachment is known as a chiasmata. That point of X-like structure, that is a chiasmata. A single crossover event between homologous chromosome, which is non-sister chromatid, lead to reciprocal exchange of DNA between maternal chromosome, paternal chromosome. So single crossing over take place between mother and father chromosome. So when that sister chromatid is moved into a gametic cell, it will carry same DNA, one from one parent and another same DNA from the other parent. So the sister recombinant chromatids has a combination of, so the output is the sister recombinant chromatid. So it consists of genes from both the mother and both the father after recombination, and it is a reason of variation in sexually reproducing organism. So multiple crossover in an arm of chromosome have the same effect. Exchange of DNA take place between the recombinant chromosome, meaning it have a new combination of genes that was not present in the original parent chromosomes. So it is a reason of variation in organism. So synaptical complex we have understood. We look at an image. Look at an image, synaptical complex, cytomeres and kinetochores where it is attached. And a synaptical complex, it's all the complex. Look at that. It is a lattice of proteins which is formed between the homologous chromosome when they are in deep love with each other. So it forms at a specific location and then it spreads to cover the entire length of chromosome. So this protein complex is present between the two lovers. So in synapses, the gene of the chromatids of the homologous chromosome are aligned precisely with each others. So the synaptical complex support the exchange of genetic materials between non-sister homologous chromatids, which is called crossing overs. Non-sister chromatid means it helps in the exchange of crossing over exchange of genetic material between maternal and paternal chromosomes, non-sister homologous chromatids. So crossing over can be observed visually after the uh, exchange of chiasmata. So kinetochore centromere image is given, kinetochore image is given, sister chromatid synaptical complex, a lattice of protein. So about that, in this there is a located at interval along the synaptical complex, there is a presence of large protein assembly, and that is called a recombination nodules. It forms at a recombination nodules. So this assembly marks the point of an enzyme uses recombinase. So these assemblies mark the points of later chiasmata and mediate the multi-step process of crossing over a genetic recombination between the non-sister chromatids. Near the recombination nodules on each chromatid, the double stranded DNA is clipped, is broken down. The cut ends are modified and new connection is made between the new sister chromatids. So as prophase 1 progress, synaptical complex begins to break down and the chromosome begin to condense, thicken. When the synaptical complex is gone, vanished, when the complex between the both partner is gone, homologous chromosome is gone, the homologous chromosome remain attached other at the centromere, they're attached at the centromere, and also at one point, that point is a chiasmata. It is also an important point, P by Q. So when the synaptical complex is gone, the complex between both the partner has gone, the homologous chromosome remain attached one place at the centromere, it is always attached and attached at the chiasmata. So the chiasmata, very important point, the chiasmata remain until anaphase 1. It is a very important point, P by Q. These chiasmata remain until anaphase 1. The number of chiasmata varies according to the species and the length of the chromosome. There must be one chiasmata per chromosome for proper separation of homologous chromosome during meiosis 1, but there may be as many as 1 to 25. As many as 25. So depend upon that length of the chromosome, chiasmata will be 1 or as many as 25. Following crossing over, the synaptical complex breakdown and the cohesion connection between the homologous chromosome, the love between the homologous chromosome is removed. At the end of prophase 1, the pairs are held together only at the chiasmata till anaphase 1, until anaphase 1, and are called tetrads because the four sister chromatids of each pair of homologous chromosome are now visible. So recombinant nodules are proteinaceous structures. Look at the image. Recombinant nodules. So these are the sister chromatid troops. These are the transverse filaments. And these are the axial filaments, axial filament, transverse filament, and it is a recombination nodules. Recombination nodules, yellow one. So these are the proteinaceous structure that form on the synaptical complex during prophase 1 of meiosis, specifically at the site of crossing over between homologous chromosomes. So these recombination nodules serve as a molecular machine which help in the exchange of genetic material during crossing overs in Pachytene stage. So it leading to genetic diversity resulting in the gametes. So recombination nodules can be visualized with electron microscope. So what are the function of this recombination nodules? It help in crossing over, it help in exchange of materials. Because of this, genetic diversity increases or happen in sexually reproducing organism. So chiasmata become visible during which stage? Diplotene, because they are separated. But only one place where the homologous chromosome are still connected, that is a chiasmata. So chiasmata is present at the diplotene stage of prophase 1 in meiosis 1, though the actual exchange of genetic material occurs during the preceding Pachytene stage. Crossing over happened in the Pachytene stage, and it's start separating synaptical complex vanished, removed, is getting lost in Diplotene stage because their love attraction between the both the couples are reduced, vanished. So this is the reason. So it's separated but remain attached at chiasmata stage. So chiasmata are a except structure. Look at image. It is except structures point of contact that hold the homologous chromosome together after the synaptic complex partially dissolve in the diplotene stage. The last stage is diakinesis. Diakinesis is after the diplotene stage, they move apart from each other, and this is called diakinesis. This is called diakinesis. Diakinesis means to move through kinesis, as you might know, movement or kinetic energy. Diakinesis, if you know the physics also, you're knowing kinetic energy. So they move apart from each other because of many fight between both the couple. So between the couple, so they start separating from each other. So finally in diakinesis, what happened? The nuclear membrane is also lost, so that in subsequent phases, that is in it is from prophase, we know about the metaphase. So in metaphase, anaphase, they could easily separated from they could be easily separated from each other. So diakinesis, the chromosome condense further to acquire characteristic size form. They separated, they condense, so the chromosome again condense. The chiasmata disappear by sliding towards the end of the chromosome, but not totally disappear. Toward the end of the chromosome, the nuclei disappear. Nuclear envelope nuclear envelope break down into vesicles. So it's become PYQ. So Pachy means thin. They align homologous chromosome, bivalents, continuous. Just it is a revision slide. It is a revision slide. Pachytene, pachynemia. So it's a formation of bivalent synapses occur, tetrads occurs, and synaptical complex formed in Pachytene. Synaptical complex formed in diplotene stage, and Pachytene, the crossing over takes place. Pachytene, stop, crossing over. Diplonema, synaptical complex formed in Zygotene, it dissolved because di means it separated. Died, just separated. So die, single entity. So synaptical complex disappear, but it remain attached at chiasmata. In diakinesis is the last stages where there the terminalization of chiasmata take place in diakinesis. The terminalization of so in diakinesis, the chiasmata terminalization take place, move to the end of the tetrads. The homologous separate further. The homologous chromosome separate further, and centromeres attach the chromosome to spindle fibers. Nuclear envelope breakdown. Dikinesis, the chiasmata terminized. It means it moved to the end of the threads. Chiasmata moved to the end of the thread. Chiasmata moved to the terminalization. Dikinesis totally separated. So terminalization of chiasmata take place. The homologous separate further, and centromeres attached to the chromosome. Centromere attach the chromosome to spindle fibers. Nucleolus and nuclear envelope break down, disintegrates. So it is about the leptotene chromatin condensed to form chromosome, zygotene, homologous chromosome come close, synaptical complex begin to form. Pachytenes, the crossing overs occurs, diplotene synaptical complex disintegrates, chiasma appears, dikinesis, homologous chromosome moves further, nuclear membrane disappears. So these are the revision slides. So it is a leptotene, zygotene, pachytene, diakinesis. Now after that, prophase 1, metaphase 1, anaphase 1, and prophase, metaphase, anaphase, telophase 1. 4 homologous interphase, then meiosis 1, daughter nuclei, 2 daughter nuclei, and then meiosis 2, 4 haploid gametes. So we are going to learn now few PYQ. Which of the following is currently represent the substage of prophase of meiosis in order? So kindly check. Synapses between homologous chromosome occurs during synapses. Synapses, we have learned synapses means there is a more love between the both parents, the homologous chromosome between both so it's a zygote. So zygote synapses with the help of, because the complex arises in zygotenes, they are loving each other, zygo with each others. So that is feeling generated. So synapses, the pairing of homologous which occurs during the zygotene stage, where the complex arises, and the complex is synaptical complex. And synaptical complex is important, which help in crossing over in later stage Pachytene. So crossing overs crossing over in Pachytene take place. Chiasmata first visible during which stage? Diplotene stage. So first upper chiasmata except structure occur in first diplotene stage. So chiasmata is except structures formed at the point of genetic exchange during crossing overs. First visible during chiasmata during diplotene stage, because they start fighting, they are separated, they become single entity, and during this stage, homologous chromosome begin to separate slightly, and the points where crossing over occurred remain connected, appearing at chiasmata, diplotene stage. So, which stage of prophase 1 is characterized by chromosome condenses and appearance of chromosome? You know very well, leptotene stage. Terminalization of chiasmata occurs in diakinesis, last stage. The chiasmata move toward the chromosome ends. Terminalization, preparing chromosome for which stage? Metaphase 1 stage. Synaptical complex is seen during zygotene stage, because zygotene generation of too much love between the both homologous chromosomes. So it form a complex, and that complex is a synaptical proteinaceous complex. So it arises in zygotene stage. In which state do homologous chromosomes begin to repel each other but remain attached at chiasmata? So diplotene. Because they start fighting, so become diplomatic, and they separated as a single entity. So synaptical complex dissolve, and it still attached at chiasmata, because no legal action has taken. The longest phase of prophase 1 is diplotene, because they are fighting, and they are trying to separate each other, but they are attached at one place, and that one place is chiasmata. Now, it's very important point in oocytes, in female oocytes, it can last for years. Diplotene stays in humans, because you learn about the female reproductive systems. It takes a stagnation for when the female child will not until it achieve puberty. So longest phase of prophase 1 is diplotene stage, especially in oocytes, immature egg cells, where it can last for months or even years. So vertebrate oocyte is the stage can be extended for years, allowing for significant growth of the oocyte. And then after, there is other process take place. I already explained this process in my video. Kindly watch that video. So human reproduction. Which of the following is not a feature of prophase 1 of meiosis? So it's look at all, and just a synapses crossing over terminalization and separation of sister chromatids. It is not. It is not a prophase 1 of, because separation of sister chromatid occur in anaphase 2. It occur in anaphase 2. Very important points. So anaphase 2, the centromere split, and the sister chromatids are pulled to the opposite poles. It pulls to the opposite poles of the cell by the spindle fibers. So diagram based question is also given, and then leptotene, zygotene, pachytene, diplotene, diakinesis. The diagram show the thread-like chromosome, just to take the leptotene stage. So diagrams show homologous chromosome paired side by side, connected by ladder-like protein structure. So zygote, that generation of this synaptical complex. So it becomes zygotene stage. Synapses occur in synaptical complex with the help of that. Diagrams show homologous chromosome completely paired with clear evidence of crossing over between non-sister chromatids. So crossing over soft. So Pachytenes, the exchange of the love between homologous chromosome will deepen, and then genetic exchange of material take place. And diagrams of homologous chromosome begin to separate but remain attached. So Diplotene, this starts fighting, and that's a single entity but remain attached at X region, that chiasmata. Diagrams of thick condensed chromosome with chiasmata at their ends and nuclear membrane breakdown. It is a last stage, diakinesis. Terminalization of that chiasmata at the end, it is going to the end of the chromosome. So it's become terminalization of chiasmata, nuclear membrane breakdown. A diagram of paired of homologous chromosome connected side by side with synaptical complex is labeled. So synaptical complex means which help in the proper pairing, and it helps in crossing over. Chiasmata are marked at several points between homologous chromosome at which stage. So we have learned. So the through crossing over occurs in pachytenes. The chiasmata are visible only in diplotene. A figure show a paired chromosome where one chromatid from each homologous has exchange segment. What does this represent? Means crossing over. A diagram show oocyte chromosome arrested for many years in the diplotene stage. What is the condition say? The condition is called diplotene until puberty. Until puberty in female gametogenesis. I explain in my female reproductive system. I already made this video. So kindly watch this video. So these are about the all about the Prophase 1. We have learned all the with the help of love between two homologous chromosome, and we have learned the mnemonics. So kindly practice that mnemonics. Practice all. I'm going to make key concept of the second video. So kindly watch that video. So we have learned in a fun way. Enjoy it, memorize it, practice all MCQ, PYQ. Thank you for watching. Watch the next series. I'm going to explain key concept related with the